The title that I prepared today is Well-Structured Strategy for Drug Development. And my name is Hyojang Kim, and I'm working in Useok University College of Pharmacy. It might be a little bit away from the pharmaceutical puncture, but I hope this can interest your scientific needs. The title today here, the title structure has dual meaning. The first meaning is literally the structural biology, which is my research field. And another meaning of structure is well-formatted drug discovery. So structural biology is my major, and this is a field to study the shape of the protein. Every protein has different figure, so by studying how they look like, we can draw their function. For example, TLR4 and GP1B, they will use their hook to grab their substrate. And the GP2B or GP3A or GP6, they will use their two big arms to embrace their substrates. So knowing the structure of protein will be a great helpful thing to develop a new drug. We can modulate their function by changing their shape. Getting to know the protein structure, there are a few methods that we can try, and the most widely and commonly used method is X-ray crystallography. If we organize protein molecules in 3D space, we can see that as single crystals. And by giving them the X-ray beam, we can have the the diffraction pattern. And by interpreting this electron pattern, we can draw the electron density map. And by fitting the known sequence into this map, we can finally get the 3D structure. There are another method that we can use to reveal the protein structure. And the other method that we are trying is NMR. This is a perfect method to see the solution structure. We can see the moving structure of the protein. So this is perfect method to see how their substrates work on protein's structure. And there are new emerging technique, which is cryo-EM. And this is actually the method that we can see how they look like in a solution. So we can use the electron microscopy to see the actual figure of the proteins. So using this method, we can use it to the drug discovery. If we have the protein 3D structure, we can see how the active site look like. And by blocking this active site with chemicals or designed peptide, we can modulate the protein's function. So when we apply this structural biology in drug development, we can reduce the drug development period a lot. So usually it used to take about 20 years to develop a single drug, but these days, using this structural biology, we can reduce this period a lot. Some AI is helping this process, and the fastest drug that discovered is some months. So I'm using structural biology to develop a new drug, and today I am going to present my projects dividing by two topics regarding their origins. So first part is about the bacterial proteins that I'm working on, and the second part will be the human proteins. And especially I am interested in the Cutibacterium acnes that causes the acne symptom, and the other interest is Mycobacterium tuberculosis that causes tuberculosis. Working with bacterial proteins has some advantages. They usually have good properties, so they are easy to handle, and the experimental process is easy, so even the beginners can join the lab work easily. I came to South Korea about 4 years ago, and in the lab setting period, the bacterial proteins were my perfect target. So among these bacteria, my interest is in Cutibacterium acnes and Mycobacterium tuberculosis, and they both have some things in common. They are really widely spread in our society, and they both are easy to get their antibiotic resistance. So my first target is C. acnes that causes the acne symptom. I think we are all familiar with this bacteria. We all have the symptom of acne in some time at our life. But in severe cases acne, they should be treated. And there are a few options that we can use to treat the acne symptom. But in severe cases, there is only one option that we can try, which is a broad-spectrum antibiotics, tetracycline. And moreover, for the last 40 years, the acne treatments has never been developed or never been studied. So I try to develop a new treatment to treat the acne symptom, and I'm focusing on the reason how the C. acnes live in our skin. So if we see the C. acnes, C. acnes lives in our sebaceous gland, and they digest sebaceous gland to get their energy, and they releases lipases, and that lipases will digest sebum lipids and resulting in the fatty acids. But when their lipase activity is too elevated, they will produce more and more lipases, and that will produce more and more fatty acids, and that causes the acne symptom. So my idea is block these lipases and use it as the acne treatment. And I figured out the structure of C. acnes lipase structure and a right side, and that has a lot in common with the typical lipase structure. They have the main domain that will perform the main function, and they also have the lead domain that will work as a protector for the main domain. The main domain has the active site, which will function as a blade of the knife, and that is covered with the pink region, which will function as a protector for the blade. And I can figure out how they work, how they open their lead domain to reveal their active site. The active site is colored in red here, and the lead opening mechanism is opening the two phenylalanine residues, changing the location of the two phenylalanine residues, and they open the lead domain to get their substrate into the active site. If I deliberately remove the gate, I can see the lipase function is elevated. So my target is close the gate forever so that this lipase cannot work as the lipase function. Since the 2 gate is composed of 2 phenylalanine residues, I came up with the idea to introduce another aromatic ring to cause the pi-pi interaction so that the 2 phenylalanine residues will not move because they were trapped with the chemical reaction. And my idea worked. I could see my inhibitor is bound to the active site, and that perfectly blocked the lipase function. So my next step is expand this structure, and then it can fit better into the active site, and then find some good delivery system, and study the animal test to develop this agent as a real acne treating agent. The C. acnes lipase is secreted to the exterior area, so it doesn't have to be drug or administration. So I've studied to develop this as a topical reagents like cosmetics or the ointment. And thankfully, few pharmaceutical companies shows interest on my project, so I'm happily working with them. So this is my first project, bacteria projects that I'm currently working on. And the second project that I'm working is tuberculosis toxin and antitoxin system. If we say that C. acnes is common bacteria in our life, this is maybe another familiar bacteria that in our lives. Approximately 33% of the human population is contaminated with this bacteria, which is Mycobacterium tuberculosis. But only few can develop tuberculosis symptom because this bacteria is really famous for their dormancy. They stay in low amount in our body, and when a bad condition, the number of them will explode. So that means that is the concept of the dormancy. And the Mycobacterium tuberculosis is the best character is dormancy. And during their dormancy, they can develop their antibiotic resistance. So it is now a big problem in most Asian countries. And tuberculosis is called as a poor disease especially in the developmental countries. So this is the one that we should defeat. This tuberculosis is really the top urgent condition that we need to defeat. So there are many theories about why they have the dormancy, but this is the well-supported theory. They do have their toxin and antitoxin operon in their DNA. These operons share the same promoter. And in normal condition, the antitoxin and toxin will be translated to proteins. And they usually bind together, and the antitoxin will neutralize the toxin's toxicity. Or the antitoxin can bind to the promoter, and they can inhibit the translation of both toxin and antitoxin protein. And with this method, the toxin's toxicity will be decreased in a normal condition. But when this bacteria gets stressed, the antitoxin will be degraded and the toxin will get its freedom. And free toxin can work as a suicidal agent to kill himself. So using this method, the bacteria can keep their quantity as low as possible in our body, and that is called as dormancy. When we compare the toxin and antitoxin operon numbers, we can see the huge amount of number can be seen in Mycobacterium tuberculosis that causes the tuberculosis symptom. So my target is targeting these 88 toxin and antitoxin system and make a new agent that kills the bacteria itself. Now, among these 88 toxin and antitoxin system, I especially target the top 10 systems that actually known as the bacteria's lifespan. So these are related with their duration of their life. So I came to South Korea in 2020. So I've been working on this project, and I've been publishing a few papers regarding this project. And my first project is the antitoxin structure with the DNA, and the other projects are toxin and antitoxin pairs. So the first target is release the antitoxin from the DNA and make the toxin protein will be translated. So by dissociating these two pairs, I can free the toxin to reduce their growth rate. And another product is dissociate the toxin and antitoxin bind structure and free the toxin, and toxin can be a suicidal agent. So I'm working on these two structures now. But all 88 toxin and antitoxin has all different figures. So they all have different shape and different binding state. So studying these other shape of toxin and antitoxin pair is another my target. So the research process is, if we have the toxin structure, the antitoxin is usually bound to the toxin, and dissociate this toxin and antitoxin pair to design a new peptide to dissociate this toxin and antitoxin pair. I need to figure out which amino acids will do the major function. And using that peptide, I need to study the binding assay using the ITC or SPR in here and design a new peptide to free the toxin. So these are my process of the research, and I still have a lot of targets that need to study. So my final goal is develop a new drug for treating the tuberculosis symptom. Like I am doing in acne projects, I need to design a new peptide and find some good delivery system and study the animal studies or clinical studies to finally figure out the new antibodies. When I say these are my bacterial proteins targets, the next one is my human targets, and I'm mainly focusing on the blood proteins. the when we say the most well-regulated system in our body, I would say the bleeding and the clotting is the best regulated system in our body. So if we have any bleeding event, there are largely three processes will happen. The first process is muscle contraction to minimize the blood loss, and then the platelet will be activated. And then next step is fibrin clot formation, which will function many clotting factors work. But but if this platelet activation stage is closely related with our cardiovascular disease, which is artery disease, so we block this process with aspirin or the clopidogrel, which is really famous drugs these days. So these two drugs will block the platelet activation. This is important because the CVD, cardiovascular disease, the artery clotting disease, is the top leading death cause in our period. So I am studying this blood clotting system to develop a new blood thinner to replace the top one selling drug in top one selling blood thinners. When we look closer of the platelet activation step, we could see the platelet will stick together using a glue, which is a von-Willebrand factor. And we can call it, in short, VWF. And that glue will be cleaved by the seizure protein, the ADAM13. And the ADAM13 only activates when VWF is activated. So I can have an idea that this protein will be a new novel blood thinners that will not cause any bleeding event, because this only activates when VWF is in inactive state. So using the ADAM13 structure, I can imagine that this will be a good blood thinner. So I published 2 papers about the structure of the ADAM13, the seizure structure. It has the blade that actually functions as a knife is in the N-terminal, and the C-terminal domain is a protector for the blade. And the structure of the blade is like that, and I can see the blade is under the bottom area. And these blades are aligned in one line so that their partner can bind easily to the blade of the seizure. So the blade should be sharpened to perform well about the seizure's function, and we figured out this structure is unsharpened blade because we could see the blockage in the linear position, and this blockage is performed like this figure. So we need to remove the blockage to make it more sharp blade. So by studying the interaction with the animal study, we could see the blade shape is changing, like there are stretched form and there are circular form. So to make this stretched blade, we can use it as a perfect sharpened seizures. And now we are studying to fix the sharpened the stretched form of the ADAM13. And another idea is blocking the protector to bind the blade. So I figured out the structure of the protector, and I could figure out how they bind together to minimize the protease activity. And my target is dissociates the protector binds to the blade to activate the seizures function more efficiently. So now what I'm working on is making a peptide that mimics the binding state and making an antibody that can dissociate the blade and the protector's binding. So these are my targets that I'm currently working on using structural biology, and I am in Usok University in Jeonju area, and our city is beautiful scenery and many cool places to enjoy. So any researchers, or we are always recruiting, so any researchers will be welcomed to my lab. And thank you for your time, and I will be welcomed with any questions. Thank you.